The genetic information that we have in our body and every other organism or even propagating entities must be some form of matter. And as we were describing, it is supposedly, it should be information. And that information should also be inheritable. So, for us to understand how nucleic acids, that is DNA or RNA, function as or can act as molecules that can carry information in the forms of bits as a sequence of subunits, we need to understand how the matter exists itself, the chemical nature of the nucleic acids, the chemistry of the nucleic acids. We will not go into very deep details as in a chemistry, but we will try to understand how the structures are. And that will form the basis for the rest of all the things. How the DNA polymerizes, how is it a sequence, what kind of bonds are involved. And then we will also know, eventually this knowledge will be useful for us in understanding many other things such as replication or even mutagenesis, and eventually also in when we study about epigenetics. I am Dr. B.C. Amar, Assistant Professor at Shastra. So the nucleic acids, like a quick review, consists of DNA and RNA. Most or all cellular organisms, starting from prokaryotes, bacteria, archaea, and highly complex eukaryotes and to complex organisms, DNA is the genetic material. In some viruses, we may have RNA as the genetic material. So some of the viruses also have DNA as the genetic material. And when we say nucleic acids, we will have three parts of it, that is nitrogenous bases and the sugar moiety and also the phosphate group. So in the chemistry of nucleic acids, I would want to remember the structure of the nitrogenous bases, the sugar, that is ribose or deoxyribose as the case may be, and also the phosphate group. And then also try to know the bonds that link these things. And then we have the formation of nucleotides. Nucleotides are the subunits of what makes nucleic acids, that is polymers of nucleotides are DNA or RNA based on the chemistry of it. We will try to, before going ahead, we will try to understand a little bit about or try to delineate how RNA and DNA are different in terms of chemistry. So the first thing is as we discussed, nitrogenous bases. The nitrogenous bases are two types, as we will see again later, but they are purines and pyrimidines. DNA has adenine and guanine as bases, purine nitrogenous bases. So does RNA, adenine and guanine. In terms of pyrimidines, we have in DNA we see T and C, that is thymine and cytosine. Whereas in RNA, we have uracil and cytosine as pyrimidines in the nitrogen bases. So the difference between RNA and DNA is that RNA has thymine, whereas DNA has thymine, whereas RNA has uracil. And then when it comes to sugar, we have a pentose and DNA has deoxyribose, whereas RNA has ribose. And we have a phosphate group or phosphoric acid, so we'll just put it as this phosphate group. In non-ionic form, you can think of it as H3PO4. Okay, so if it is an ionic form, you will have two less hydrogens in ionic forms. Yes, so these are the differences. So primarily the difference between RNA and DNA in terms of bases is one methyl group. Just try to remember it for this moment. At this point, the difference in the chemistry between thymine and uracil is one methyl group. And in terms of ribose and deoxyribose, the difference is deoxyribose has one less oxygen. That is the only the difference between the DNA and RNA when it comes to at the level of elements or groups. We will see those slowly now. So first things, purines, and the purines are adenines and guanine. So I will try to draw the structures and try to derive it. The purpose of drawing the structure is that you will be able to also follow the hand movements, and it's important that we practice multiple times so that it will become easy for everybody. So I'm trying to draw the structure of a purine. And this is the structure of purine, which would have double bonds, something like this, and then we do have hydrogen atoms here and there to balance out. So there is 6 amino purine. So we would probably also discuss the numbering here itself. So the numbering for purines goes this way: 1, 2, 3, 4, 5, 6, 7, 8, and 9. So the numbering for purines is such a way, and for adenine, let's check. This is adenine, and I'm going to repeat the structure as it is. Adenine is something like this, and 6 amino purine is adenine. So if we recollect the numbering of it, it would be 1, 2, 3, 4, 5, and 6, 7, 8, 9. So all that is different is that if I add one amino group here on the 6th carbon position of 6th carbon, then 1, 2, 3, 4, 5, and 6. So we will have these the double bonds wherever it should have been. Say that roughly that is the structure for pyrimidines, and from there we can derive the 3 bases. For me, I would go with uracil first. Uracil is present in RNA but not in DNA. Uracil structure would then be N this way, and you have oxygen there, and oxygen, I think it's important to note the numbers first: 1, 2, 3, 4, 5, and 6. This is going anti-clockwise. So if you want to describe uracil, we can put it as 2,4-dioxy pyrimidine. So we have to make up these names so that we will be able to remember them for a longer time, and that will become important. Okay, so then we will try to bring about thymine. For me, thymine is easy because thymine goes this way. Thymine is 5-methyl uracil. We have already derived uracil, and now we just have to add a methyl group, and that becomes thymine. So this is uracil. For uracil, I should have put the double bonds there. Okay, now we have 5-methyl uracil is thymine, and the numbering should be there. It would be 1, 2, 3, 4, 5, and 6. And on the fifth carbon, I should add a methyl group. And that makes it thymine. This is uracil, and this is thymine. And now we will work out for cytosine. Cytosine is almost similar. We have to develop our own ways of how we want to remember it. So it is almost similar to that of uracil, except there is some change. That is, uracil has, let me write the numbers again. Numbering 1, 2, 3, 4, 5, and 6. So on the second position, oxygen is similar to that of uracil. What is different here is that on the fourth position, there is an amino group. And the rest of the things will be same. Okay, so I hope you can remember these. I have tried to draw the structures in a way that is as a scheme from which you can derive one from the other, and that is a good way of remembering things. I would have gone with these structures of pyrimidines and the purines and pyrimidine structures, and I should have remembered the numbering scheme. From then on, if it is 6-aminopurine, it is adenine. If it is 2-amino-6-oxypurine, then it is guanine. And if I have 2,4-oxy-dioxypyrimidine, then it is uracil. If I have 5-methyl-uracil, then it is thymine. And if I have 2-oxy-4-amino, then it is cytosine. And that is a good way to remember, so that you will recollect. The numbering is important because often we do speak in for example, damage in DNA happens, and then we do discuss about some of the groups have lost, and we might mention the numbers. So there it might become important for us to remember. So next is the simpler ones that we need to remember is ribose and deoxyribose. So ribose is simple. We draw it this way, the boat structure or ring structure. And then we have, say, OH, H, and then CH2OH. So in this case, because we are talking in nucleotides, we have already assigned number of positions 1, 2, 3 to the ring structure or the nitrogenous bases. The numbering of sugars will also have, similar to that, we will also have the numbers here. This is 1 carbon, 2, 3, 4, and this is the 5th. Now, I hope you recollect that the nitrogenous base and will be attached to the ribose. And if I were to say it is 2 carbon of something, am I referring to the base or am I referring to the sugar? There will be a confusion. So to avoid that, we put 1 prime, 2 prime, 3 prime, 4 prime, and 5 prime. Every time we say 1 prime, we are mentioning about the carbons or the positions of the ribose sugar or deoxyribose. If I do not say 1 prime, then it is meaning that we are referring to the nitrogenous base. So ribose is easy, and deoxyribose is, for deoxyribose, it is 2 prime deoxyribose. So that means we would have everything very similar to that of ribose that we have drawn. And let me put the numbering, that is 1 prime, 2 prime, 3 prime, 4 prime, and 5 prime. Many times we would find several people describe referring it to as 1 dash, 2 dash. I think that is wrong. I think 1 prime is correct, but it's up to you. So 2 deoxy we are saying, otherwise we had OH here, H here, and this is OH. We also had OH on the 2 prime for ribose, as in this case here. But we do not have this. Instead, we just have the hydrogen. So the oxygen is not there. That is why it is deoxyribose. So this forms the deoxyribose is present in DNA, whereas ribose is present in RNA. So we need to now try to make the compounds, and I think this is something where we will discuss a lot of these together. So we have a nitrogenous base. Nitrogenous base will form a bond with the ribose sugar. So let's draw adenine again. N. So we have OH here. Deoxyribose is 1H in a 2 prime position, and we have OH here, and this OH and the hydrogen with the nucleotide, that is thymine, will be released out or will come out as water molecule, and then there will be a bond formation between the first carbon and the nitrogen of 1. So that is the new glycosidic bond that has formed. So anytime we call these alone as nitrogenous bases, this one, when it is coupled or when it is bound through a glycosidic bond to the deoxyribose or ribose, as the case must be, then we call it as nucleoside. And this alone would have been the nitrogenous base. So I hope we will just quickly have a review of it, and then we will move on. So purines are 2, adenine and guanine. They both are common for DNA as well as RNA. The numbering should be, if we remember the structure of purine and numbering scheme, then everything else is easy. From there, 6-amino purine is adenine, 2-amino 6-keto is guanine. Purine, 6-keto purine is guanine. Similarly, pyrimidines, they have a single ring structure like this, And if you remember uracil, that will be easy. In the purines, the numbering system starts with the N here, and then it goes clockwise. And in the pyrimidines, the numbering starts with this nitrogen here, and it goes anti-clockwise. And if you remember the pyrimidine structure, from there we can get uracil. It is 2,4-keto or dioxy pyrimidine. From there we can get to thymine. It is 5-methyluracilase thymine. And then we can also get cytosine from here, replacing the keto group on the 4th position with an amino group, and that is cytosine. And then the base, nitrogenous base, can be bound with the ribose or deoxyribose, as the case must be. The difference between ribose and deoxyribose is here in the 2nd position. That is, OH is present in the 2nd position for ribose, whereas 1 oxygen is less for deoxyribose. The numbering system also is important for when we trying to recollect about the nucleotides. The first carbon, 2nd carbon, and we indicate with a 2' or 3', something like that, to denote the positions of atoms in the sugar, especially when we are talking in terms of nucleic acids, so that we can distinguish between the positions of nitrogenous bases from that of the sugar, or the ribose or deoxyribose. So when the base is bound by a glycosidic bond with the nitrogenous base, we call it as nucleoside. If the nucleoside is bound with a phosphate group, then we call it as nucleotide. It is usually NMP, nucleotide monophosphate or nucleoside monophosphate. And nucleotides could be in 3 different ways, at least, based on the number of bases, the number of phosphates that are attached to the 5' carbon. So when we are talking about nucleic acids, there are 2 important positions, that is 3' and 5' of the ribose or deoxyribose, as the case must be, because they are involved in the polymerization to form longer DNA or RNA. So there are phosphate groups. One, it is monophosphate, nucleoside monophosphate. If there are 2, then you call nucleoside diphosphate. If there are 3, you would call nucleoside triphosphate. Say, for example, if it is adenine and we have, say, deoxyribose, then it is a nucleoside. Deoxyribose plus 1 phosphate group, here it is A, so it is called as adenosine monophosphate. If we have another phosphate to it, then it is ADP, adenosine diphosphate. Again, I said deoxyribose here, therefore it must be deoxyadenosine monophosphate, and here deoxyadenosine diphosphate. And you can add 1 more phosphate to get the deoxy ATP. If this sugar is just ribose, then we would have called it as AMP, ADP, and ATP. I hope this is a quick review of all the things that we have learned. There is one final point that we need to also remember in terms of phosphates. The phosphate that is closest to or directly attached to the sugar moiety, ribose or deoxyribose, is denoted as alpha, and the next one is called as the beta phosphate, and then we have the gamma phosphate. So the numbering here starts from closest to the ribose or deoxyribose and towards the outside. Thank you very much.